All right, today I'll be talking about mRNA localization and specifically its experimental history in budding yeast. So first we want to look at the ASH1 gene. And this gene is pretty essential for separation of the daughter cell because you can see that this daughter cell requires the localization of this mRNA to its distal tip. If you see that, oops, let me get my... If you can see that, right? This is the daughter cell right here, and you can see that ASH1 mRNA had to go there, and this is to suppress mating type switching. We don't really have to know what this means. We want to look at the molecular basis for this localization. It was found that this myosin protein, myo4p, was required for localization. And so this kind of suggests that the actin skeleton is probably very important for this localization specifically. Now, we also found that mRNA was not localized in an actin mutant, so this sort of confirmed this. And so we now know that we have a bunch of actin filaments, and myo4 will bring our mRNA. Oh my god, this laser pointer is bad. This is our actin, this is our myo4, and then our mRNA, right? And then this guy kind of walks to the distal tip of the daughter cell. That's sort of what we know so far. But how does this interaction work, right? I mean, is it myo4 directly binding to the mRNA, or are there other proteins involved, And so what researchers did was they found cis-acting elements that bound directly, that were required for localization. And so they did this by fusing, we see this is the ASH1 mRNA, as well as a gene that was not localized. It doesn't show it here, but you can kind of see the fusions. Basically, the parts here where we can see like the outline is the part where we took it from the ASH1 gene. And the empty spots, right, all this empty spot is from the other mRNA. And so if we only had this part that was specific to ASH1 gene and we saw localization, that suggests that a signal in that section is important for localization. And we can kind of see all the charts here. And by overlaying everything, they could find that specifically E1, right, E2A, E2B, and E3 were the ones that were the most important. And they were also redundant, right? So you only needed one for localization. You can see, you can examine this graph, right? E1, if you draw a line down here, every single localized mRNA that didn't contain the other three sequences, right, had to have this entire sequence. You can see that 26, right, only has like a bit. They lost the front here, and that wasn't enough. And so they found that the smallest possible section of this mRNA was just this little part for E1. And then the same thing for the rest, E2A, E2B, and E3. And you just need one of these for localization. So this was very important in determining where exactly on this gene is like the localization signal, if you will. And so they also found another protein called SHEP2, or She2P, but I'm going to call it SHEP2. And to confirm that this did play a role, they basically used GSD labeled SHEP2P, SHEP2, and then they used a UV cross-linking assay. And what they did was they took unrelated RNA that was labeled, right, labeled RNA. And so obviously if you have unrelated RNA, you're still going to get binding of SHEP2, right? Because SHEP2 will have just like, random binding, like it's fine, right? And we're specifically looking for mRNA that contains SHEP2. So you can see this here. They label this in kilodaltons, but just know this is mRNA signals. Okay, this is not protein signals because we're running a UV cross-linking assay, right? Remember, we have our RNAs, we have our proteins that just bind, and then we cut them with RNases, right? And then the ones left will be the ones protected by proteins. And then when we undo the cross-links and run them on a gel, right, we'll see exactly the RNA sequence on the, let's say, acrylamide gel, right? And what they found was that as you increase the concentration of unrelated RNA, you can see obviously everything increases, right? You're getting just a lot of random proteins binding and protecting some RNA, right? However, when you added E3, remember E3 was just this little part, right? This little part of the 3' UTR that we did see was involved in localization. We can see that SHEP2 bound pretty tightly, right? Because, I should say, this E3 unlabeled, right? And so it wouldn't show up on the gel, right? Remember, the unrelated RNA was labeled. Whereas the E3 was unlabeled, and so as you added more E3, we can see that SHEP2 binds more, and we can't see anymore, right? Because it's bound to the unlabeled RNA. Whereas, the labeled RNA is still there before you add E3. So it suggests that there is, competitive binding for E3 of SHEP2. And they saw this was the same case for all of the other binding sequences too, which kind of makes sense. Though they were different affinities. You can see that E2A has a much smaller affinity than E3 or E1 or even E2B, right? But E2A is still, it still competes for binding compared to just, random RNA, right? That's sort of what we saw. And this is, remember, this is the RNA that's showing, not the protein, right? The RNA is showing. And what's showing is the unrelated RNA. Unrelated RNA. And what's not showing anymore is the unlabeled E3. Okay? And so if it decreases, right, that means there's more SHEP2 bound to unlabeled E3, meaning less SHEP2 to bind to the unrelated RNA, and therefore there's a smaller band. And obviously, the sizes of these bands, the reason why we can distinguish them is because how big SHEP2 is will determine how big the subsequent protected region would be, right? And so scientists could figure out that this was SHEP2. You could use a DNA ladder or whatever, right? Or some controls. So how does SHEP2 exactly recruit this myosin protein to ASH1, right? Because now we clearly see that there is some connection between SHEP2, ASH1, and therefore myo4P, right? And so what they did is they first found another protein. All right, stick with me. SHEP3, right? That was also required for mRNA localization. And bear with me, this will make sense in a bit. And what they did was they tagged with GST both SHEP2 and myo-P, okay? So both SHEP2, I'll draw here, SHEP2 has a GST, and same with myo4P, okay? And these are bound to glutathione, glutathione, whatever it's called, right? This guy, right? This guy. And this is bound to a bead, right? This is a bead. And then basically, if you're mixing together with radioactively labeled SHEP3, right? If SHEP3 were to bind, and there's another one, it's just GST on its own, right? So if SHEP3 binds to SHEP2 in myo4, then it would bind here, right? This would be SHEP3, and it would bind here. And then we could see it, right? We could see these beads. And so that's exactly what we saw. And what we also saw was that GST on its own, right, this is our negative control, didn't bind, right? There's no binding here. So this confirms that SHEP3 bound to both SHEP2 and myo4P. And other experiments also show that SHEP3 binds to different parts of SHEP3 binds to SHEP2 and myo4. So this kind of suggests that it's maybe an adapter protein, right? So maybe that's the link, right? We have our mRNA, and obviously we know that SHEP2 binds to mRNA, right? I'll just show this next slide, right? We know that SHEP2 binds to the mRNA, and now we have SHEP3, right? That kind of acts like the adapter intermittent zone, and then we have myo4 that's obviously on the actin filament to bring it to the distal tip, right? And so this kind of suggests that if we could get SHEP3 to bind to the mRNA on its own, then we could localize the mRNA anyway, right? This is sort of what this model would predict. And so if we want to test this model, we got to see, okay, if we remove SHEP2, but we somehow get SHEP3 binding to the mRNA, do we get localization? Because if so, that really supports this model. And that is exactly what they saw. So what they did was they made these MS2 coat proteins fused to SHEP3, right? So we have SHEP3, and we have this MS2 coat protein bound to it, right? This was what they made. And this MS2 can bind to a specific sequence on an RNA, so long as we make the RNA, right? To have an MS2 binding site. And so then in this case, right, these guys should bind, and therefore this kind of lets us have, or you can see it in this diagram, right? We have our MS2 binding site that would bind to the MS2 coprotein, which is fused to the SHEP3, and SHEP3, as in our model, should bind to MYO4, right? And this should result in localization, and that's exactly what they saw. They saw localization, and this was independent of SHEP2. So this confirms our model. This really supports our model. And so we can safely assume that SHEP3 is an adapter protein. And so now they just kind of wanted to see, how this looked in vivo. So they made two constructs, right? One was a GFP fused MS2 coprotein, just like the ones we saw before. And then we had an mRNA with a bunch of MS2 binding sites along the ASH1 gene, right? And so this gene contained E3, so this should localize. And so if we imagine what this looks like, right, is we have GFP. Remember, this is green fluorescent protein, so we can see this. And this is bound to MS2, right? And MS2 will bind to our mRNA, right? And this mRNA contains the E3 ASH1 3' UTR. So this guy should, bind to what? SHEP2 and then SHEP3 and then MYO4, and this guy should localize. And that's exactly what they saw. They used live cell imaging, and basically what they saw was, let's say this is the cell, and let's say this little red part, or this green, right? this green part, right? They saw that it slowly made its way to the tip. Whereas for the MYO4P mutants, right, meaning that MYO4P just couldn't bind, right? They saw completely random diffusion, and they didn't really go anywhere. Okay, so that's just kind of cool. They kind of saw this in vivo, saw this localization in action. But now they want to go back. Okay, let's look back at SHEP2. Okay, let's take a look at its role in nuclear export, right? Because if it's moving this mRNA, right, or it's involved in moving it, is it like involved in nuclear export in some way, right? What's going on? So what they did was they used the temperature-sensitive MEX67 gene. Okay, and this gene encodes for proteins that is important in nuclear export. Okay, and then they tag SHEP2 with an epitope. An epitope basically is any sequence, right? Amino acid sequence that binds to an antibody. Okay, that's basically what an epitope is. And so we could, antibody this guy. We can get an antibody to bind this, and we can like immunoprecipitate, or we can immunofluoresce, and just to visualize. And that's what they did in this case, right? And what they found was that in a permissive temperature, right, SHEP2 is found outside at the distal tip. Right, if you take a look, you can see that it's at the distal tip. Whereas on the non-permissive temperature, it kind of just stays inside, right? It's kind of just chilling in the nucleus because, well, it can't go out, right? And so just to suggest that, okay, SHEP2 is found in the nucleus to begin with, and then it directs nuclear export, right? But obviously nuclear export has to be allowed first, which is encoded by this MEX67 gene, which is turned off in this case, and that's why it stays in the nucleus, but it's turned on at a lower temperature, which is why it's at the distal tip, right? That should make sense. All right, now we have a bunch of series of experiments, and low-key, this is like the most tricky part that, was kind of confusing during lecture. So first, they found that SHEP2 associates with both the mRNA and the gene itself. Okay, so it's not just an mRNA; it can also associate with like the chromatin itself. And unsurprisingly, they use chromatin immunoprecipitation, right? They use CHIP, and what they measured was transcriptional enrichment compared to untranscribed regions of the yeast genome. Right? So if you have a higher level, this means more transcription. Lower level, less transcription. And what they found was that there was no enrichment for both the promoter and the N-terminal coding region of the mRNA, which kind of makes sense, right? There was, if we go all the way back, like nothing here is gonna be like, no one cares about that, right? Remember, this is chip, right? So we're basically using like we're cutting it up, and then we're looking at the protected regions, right? And we're seeing how much of it there is. But we see a lot of it, right? A lot of it in E3, E2, and E2 and E3, right? E1, E2, E3. And this is just SHEP2 tagged with MIC epitope, right? And we can see there's a lot, which makes sense, right? That's nothing new. But what's interesting is that after you add RNases, right? And if you add RNases, you're gonna break up all of the mRNAs. And so there wouldn't be any binding. So all the binding you would get left wouldn't be from the mRNAs, but we still saw stuff, right? We're still seeing stuff. And this can only be explained if it bound to our chromatin, the genes themselves, right? That's the only way we could see this, right? Because we destroyed all the mRNA, but it's not, the RNases are not going to destroy the chromatin itself. And after we run CHIP, right, we're going to cut those guys up, and then we're going to immunoprecipitate them, and that's what we see. So it suggests that, okay, SHEP2 associates with both the mRNA and the gene itself. Obviously, mRNA at a higher extent. But we want to see exactly how much of a higher extent this is, right? And so they found that, okay, ASH1 must be transcribed for a very efficient association, which kind of makes sense, right? If we take a look at here, right, what they did was they had the same, reagents, right? You had SHEP2 on its own and the SHEP2 with RNAs, right? And what they did was they made ASH1 gene fusion that was under the control of the promoter that's inducible by galactose and repressed by glucose. So you can imagine lac operon, right? Imagine a lac promoter region where you have, like a cap binding site as well as a lac repressor binding site, right? And so you add galactose, it's going to get induced. If you add glucose, no cap binding, right? So there's not going to be transcription. And what they found was, and they did same chromatin immunoprecipitation in either you add glucose or you add galactose. And glucose should obviously repress, galactose should obviously transcribe, right? And we see that there is very similar binding, like adding glucose. So if you repress transcription, you see very similar binding, which makes sense, right? That kind of also just means our RNase works, right? Our promoter thing works. But after you add galactose, there is a huge jump. So this is kind of just like supports what we saw up here, right? But now we kind of confirm it that yes, it binds to both mRNA and the gene itself, but clearly the mRNA is a lot more important. But interestingly enough, SHEP2 actually binds first to the ASH1 gene. And the way they did this was they used a time course experiment. So what they did was they, use the same construct up here, right? So it's inducible by galactose. And what they did was they added the construct, right? And then they added galactose and they started a timer, right? And then after like a certain amount of minutes, right, they looked at the fold enrichment just like the same with CHIP. Except now they're just looking for either the gene with SHEP2 or the mRNA with SHEP2. And remember, we could do that using this method, right? We can add RNAs to just look at the genes themselves, right? Or we can just not, right? And then we can look at that. What they basically found was this mRNA plus SHEP2, which would be this line, also contains gene binding. That's very important because they can't really differentiate that, right? You can't really just destroy the DNA. Well, I guess you could, but like they didn't, right? So importantly, this also contained the genes. This only contained the genes, right? So the top here, the ASH1 gene is just the genes, but the ASH1 mRNA also contains the genes. Okay, that's a little important. And so what they did was they found that it actually binds to the gene first. You see this? It binds to the gene first. And then, after a while, does it start binding to the mRNA, right? And even the mRNA, a lot of this is also still the gene. So this is very supportive. That look at that. That's a pretty big time, right? Big time difference. SHEP2 first binds to the ASH1 gene. That sort of suggests that ASH1 binds to the gene first, and then as this is ASH1. Oh, no, sorry, not ASH1. That's SHEP2. And let's say they're transcribing the mRNA. Then it kind of hops on, right? That's sort of what it's suggesting, right? It suggests that it first, huddles around the gene first, and then it binds onto the mRNA. So that's just, more molecular characterization of SHEP2. And so they wanted to take a closer look at this, right? They wanted to see, okay, is this really true? Because if this were true, right, then SHEP2 should have some nice binding with RNA polymerase, right? It should be good friends with RNA polymerase. Well, let's see if that's the case. So what they did was they used co-immunoprecipitation. Now, basically what that is you have an antibody, and on the ends of the antibody you have SHEP2 or whether protein 1, but in this case it's SHEP2. And obviously this can be precipitated using the proteins that are bound to it. And then whatever precipitates up here, right, whatever binds to SHEP2 will be co-immunoprecipitated, right? Because we're just precipitating SHEP2. But whatever is bound to SHEP2 is also going to get carried along the ride, right? So that's essentially what they did here. They tagged SHEP2, okay, that's nothing new. And then they co-immunoprecipitated all the proteins, and then they ran it on a gel and looked at it with Western blot. But they specifically also used an RNA polymerase antibody. So if we imagine here, the input is just all proteins, okay? This is not specific at all. You just have this is kind of like our positive control, if you will, because every single protein in here, RNA polymerase has to be in there, right? You can still also see this is just a random protein that they use to control for specificity of binding, right? That'll be important later. But then this IP section, they only use the co-IP'd proteins, right? So only the ones that bound with SHEP2. Okay, that's all they had in this second lane. And they found that, okay, RNA polymerase was there. Great, RNA polymerase was there. And there was no PGK antibody lighting up. And that suggests that this was at least somewhat specific for RNA polymerase. And to make sure that it actually bound to RNA polymerase itself and not just the mRNA, right? Can you imagine mRNA could also bind to SHEP2 and RNA polymerase could be there. They also added RNases, so this would be cut. So in this case, there would be no RNA polymerase bound if this was the case, but they still saw it. So that sort of confirms that yes, SHEP2 and RNA polymerase form some sort of complex. That's even more important. That's very key. And then the last one, this was just like an extract that didn't have any SHEP2, so obviously it's not going to immunoprecipitate in the first place, and so this is sort of our negative control, right? And so basically the bottom line is they found that RNA polymerase and SHEP2 bind together and form a complex in this mechanism. And that's very important because now we know, okay, ignore this CEP4, CEP5 for now. We know that SHEP2 and RNA polymerase are friends, right? They're friends. And then after mRNA is transcribed the SHEP2 jump, remember we found that SHEP2 bound to the gene first and then the mRNA, right? And now we kind of see that it was actually just because SHEP2 chills with RNA polymerase. So that's pretty cool, right? It could also be on the gene, but maybe it hitches on polymerase 2 when it approaches, right? Who knows? But either way, the important part is it's with polymerase 2, and then once the mRNA is transcribed, it jumps on mRNA. Great. But to go back to this SPT4 thing, they found that this also co-immunoprecipitated with SHEP2, so they also bind. And in their absence, there was no SHEP2 recruitment. So this is another layer of adapter protein, importance, right? Because maybe CEP4 and CEP5 are required for SHEP2 association with RNA polymerase. And so this is what they asked, does it play a role? And they found that it does, actually. So what they did is they had an SPT4 mutant, right? So this was non-functional. So wouldn't be able to form that SPT4-SPT5 complex. They also just had the wild type. And then they had a PAF1 mutant, which was not involved with ASH1 mRNA at all. So this is our negative control, right? And so they saw that in the absence of SPT4, there was inefficient localization. And you can see that here, right? The way they did it is that they separated the distributions of all of their samples into 3 categories of localized, but localized, and delocalized. right? So localized is not particularly specific, right? It can be localized anywhere. But localized is good. This is what we this is what would be pretty good. Delocalized, on the other hand, is bad, right? Delocalized, that means that it didn't work. Nothing worked. What they found was that in the localized section, we had a lot of wild type and a lot of the other random mutant, right? This makes sense because, well, this is just normal localization. But with the SPT4 mutant, not much of it was in the localized section. In fact, it wasn't really in any section. It was kind of just like all around, right? It's a little in the bud localized, so this suggests that it kind of still does something, right? But at the same time, it's not great because, and also if you take a look, bud localized also has a bit of wild type and path 1. The reason why they're not also high is because this is a measure of proportions, right? This basically just tells you the majority of wild type is here. And then there's equal amounts of bud localized and delocalized. This is just like random proteins, right? This is obviously going to happen in biology, right? But most of it is localized. That's what's important. And we can see that most of the SPT4 is delocalized. And this is just indicative that, okay, this is required for localization because most of it is actually in the delocalized distributions, right? And so, yeah, this is just a crucial role for SPT4. And so that's where we get to our final model. SHEP2 chills with both SPT4, SPT5 complex, as well as RNA polymerase. Everything here is essential. SHEP2 will bind to the mRNA, right? And then once it binds to the mRNA, SHEP3 can come in. And then we have our myo4, right? And then we have our actin filaments, and myo4 can walk towards the distal tip. That is our final model, and this is how we get ASH1 localization to the distal tip of our daughter cell of our yeast. And yeah, that is, in summary, what mRNA localization is and its experimental history.